I will begin by carefully removing the spent culture media from the T-75 flask. Using a sterile serological pipette, gently aspirate the media without touching the cell monolayer. This step removes metabolic waste and serum proteins that can interfere with trypsin activity. Once removed, discard the media into the appropriate liquid waste container. Next, I will wash the cells with phosphate buffered saline or 1x PBS. Add 3 milliliters of sterile PBS to the flask, making sure the entire surface is evenly covered, and repeat this twice. Gently tilt and rock the flask to wash away residual serum and dead cells. Avoid shaking too vigorously, as this may detach the cells prematurely. Once the wash is complete, carefully aspirate and discard the PBS dot. Now, I will add 5 milliliters of trypsin to the T-75 flask, and the volume of trypsin depends on cell line and confluence of cells. Make sure the trypsin evenly coats the entire surface of the flask. Trypsin is an enzyme that breaks down adhesion proteins, allowing the cells to detach from the flask surface. After adding trypsin, gently rock the flask once or twice to distribute it evenly. Now place the flask into the incubator and allow it to incubate for 5 minutes at 37 degrees Celsius. After this time, check the cells under the microscope if needed. The cells should begin to round up and detach from the surface. Avoid over-trypsinization, as prolonged exposure can damage the cells. After incubation, remove the flask from the incubator. Gently tap the sides of the flask to help dislodge any remaining attached cells. Using a sterile pipette, transfer the cell suspension into a 15 milliliter falcon tube. To stop the action of trypsin, add 5 milliliters of complete growth media to the falcon tube. The serum in the media neutralizes trypsin and protects the cells. Next, place the falcon tube into the centrifuge. Centrifuge the cell S for 5 minutes at an appropriate speed, typically around 300 G. This step pellets the cells at the bottom of the tube, allowing removal of trypsin and old media. After centrifugation, carefully remove the tube without disturbing the cell pellet. You should see a visible pellet at the bottom of the tube. Gently aspirate and discard the supernatant, leaving the pellet intact. Finally, resuspend the cell pellet in 5 milliliters of fresh complete media. Gently pipette up and down until the pellet is fully dispersed and the suspension appears uniform. The cells are now ready for counting, passaging, or further experiments.